Thank you very much for introduction, and I don't have COI. So upon the outbreak of COVID-19 virus infection, and I said people were very concerned about immunological function of food, diet, and also supplement. And today I would like to introduce about the potential immunological function of chlorella by considering the effect on the immune cell by a pattern recognition receptor, and also the potential effect on immune cell by a gut microbiome. So chlorella is eukaryotic and unicellular green microalga at size of 5 to 10 micromicron. It grows in fresh water and proliferates very rapidly, and one of the most commercially used microalga as food supplement. The source of, and actually this chlorella is the source of valuable chemical for biofuel and also potential nutraceutical. And this is a kind of the systemic taxonomy indicated. And chlorella belong to phyla, the chlorophyll, and I think this is also belong to two classes. This Tohoku and chlorophyll. And importantly, the majority of the food industry use the two different species, Chlorella vulgaris and Chlorella pyratoidosa, but this is based on the relatively classical taxonomy. Recently, the taxonomy of the chlorella has been revised, and several researchers started checking about also the species, and indeed these two species used in the many industry and research would be not only two, and it contains many types of different species. So we need to think about furthermore in the future, think about the species in new name. But in any case, the majority of the food research still in use this in all the taxonomy. So this is the structure of chlorella, and when we think about immunology and immunological function, for us as an immunologist, it's very interesting. They have this rigid cell layer. And this is the study by the Austrian group that they checked that 13 different species of chlorella, and they showed that the composition of the sugar could be relatively different in each group. And also, this is a gathering of the data that shows the structure of the chlorella, and it shows that chlorella actually has a very heteropolysaccharide. And the first and the relatively first study done in 2005, and they say that arabinoglucan could be the structure, but actually indeed there are very different types of sugar could be contained in the cell layer. And a recent study also shows that arabinoglucan structure in the Chlorella vulgaris could be relatively similar to the lipoarabinomannan from the Mycobacteria. And the LCMS study, this is also by the Austrian group, shows that the Chlorella vulgaris are oligomonocyte and highly methylated at the N-glucan type. So then, thinking about the different type of the kind of structure or carbohydrate contained in the cell layer of chlorella, we can think about perhaps this could bind to the pattern recognition receptor, which can bind to the molecular pattern associated with microprobe, which express in the cell surface of immune cell, in particular macrophage and dendritic cells. This is a scheme, actually for Toda receptor. Toda receptor are the representative receptor for this pattern recognition, a receptor expressed on the surface of the macrophages. And for instance, Toda receptor 1,2, whereas Toda receptor 2,6 and Toda receptor 4 can bind polysaccharide. And the presenting cell also express various types of C-type reaction receptor, which can bind also polysaccharide from the foreign source. So then question could be raised, chlorella does have immunological function. Seems to be yes. And then this is a kind of the list from the old study. And actually, the majority is based on the mouse study, but this is a report about anti-tumor immunity, what also improves the function. And some researchers said this could be by Tohoku receptor 4. And the stimulatory effect can be really observed in some paper, and the anti-allergy effect, and so on. And then we are also interested, and the Chlorella can stimulate dendritic cells and macrophages. So we did some experiment. And we prepare for the extraction from the Chlorella pyrenoidosa, which was provided by some Chlorella. And then make the fraction, and this is water-soluble fraction. And then, depending using the filtration chromatography, we make 1, 2, 3 for fraction. And we use first a mirror macrophage strain, 2, 4, 6, 7, and stimulate them with this fraction. And what we found is that, and this is not so easy to tell, but I think that the red line is the data of the fraction sample, and the blue line is the positive control, the LPS stimulation. And we found that fraction 1 induced activation of this strain relatively significantly, like LPS. And we use the Tohoku receptor 4 inhibitor or neutralizing receptor Tohoku receptor 4 and Dectin-1 inhibitor. And what we found is that neutralizing antibody against Tohoku receptor 4 abolishes the effect of the Chlorella extract in the activation of the membrane macrophages, which means that Tohoku receptor 2 could be involved in the activation of the macrophages. And we use also the bone marrow-derived membrane dendritic cell and stimulate the fraction 1, and we found that interferon beta, type 1 interferon, and IL-6 and IL-10 production could be observed in the stimulated cells. And type 1 interferon is very important for the antivirus protection. And the IL-6 is called pro-inflammatory cytokine, but it's important for the antibody production, and the IL-10 is anti-inflammatory cytokine, which reduce the excess or undesired immune responses. This is good in combination. And in the co-culture system with the dendritic cell and T cell, we found that in the presence of the Chlorella extraction, the T cell cytokine production is enhanced, like IL-2, IL-17, and interferon gamma production is actually enhanced. And interferon gamma is produced by Th1 cells, and the IL-17 cytokine is produced by Th17 cells. And Th1 is a very important protection against the intracellular bacterial infection, and the Th17 is very important against the infection, against protection from the extracellular bacterial infection. And then the dendritic cell can produce type 1 interferon. This is important for the virus infection. So it seems to be that the Chlorella seems to have the potential of the immunological function. And then we are really also interested in the effect on the immune cell by a gut microbiome, and Chlorella could perhaps affect also gut microbiome and then induce some kind of immunological function by this function. Then, and I think we noticed that fraction 1 would be immunostimulatory molecule compound, and then we perform, actually our collaborator performed GCMS study, and he checked this fraction 1, and indeed this contains a lot of the carbohydrate, and then, and they contain also arabinose, and glucose galactose seems to be non-digestible carbohydrate, and importantly, non-digestible carbohydrate could be utilized by gut microbiota. And then we are also interested cellular extract could be used, right, for this gut microbiota for the beneficial effect. And then this is the data incubation of this Chlorella fraction with lactobacillus acidoporphyras. This is mostly found in humans, especially in the gut intestinal tract, and used as probiotics. And then what we found is that fraction 4 and fraction 1, and this is not non-fractionalized, the crude product, can proliferate, induce the proliferation, or the growth of this lactobacillus. And this means that the Chlorella can be also used as a prebiotics. And for us, it's interesting that this fraction 1 can be the immunostimulatory or functional fraction, and also at the same time that can induce the growth of this lactobacillus beneficial bacteria. And we can think about how it can affect on the gut immunity. So a microbiome can digest I think that the human cannot digest. That's why we call this non-digestible polysaccharide. But microbiome can digest and produce various types of metabolite. And that representative metabolite could be short-chain fatty acid, such as acetic acid, propionic acid, and lactic acid. And this short-chain fatty acid can induce, stabilize the FOXP3, which is important for the regulatory T cell differentiation. So which means that these non-digestible polysaccharide can modify or induce really the kind of the non-inflammatory condition in the gut. And more importantly, or also similarly importantly, the B cell can receive this signal by the short-chain fatty acid, can differentiate into plasma cell, which can induce the IgA production. IgA is very important, the protection from the virus infected in the mucosal layer. It includes the COVID-19 and influenza virus. So indeed, if we check the publication, several studies show that intake of product, and this is the data from the Chlorella planoidosa, increase the salvia IgA level in human adult. And this is an in vitro fermentation study by showing that the Chlorella planoidosa extract increase the level of short-chain fatty acid in vitro fermentation using stool from healthy and celiac donors. And this is the data upon the stimulation or the incubation with the Chlorella extract. I think this is N1, so it's not so conclusive, but it is indicator potential. And this is a randomized double-blind placebo-controlled crossover trial, including 40 Japanese participants with constipation. And this Japanese researcher group shows that the subject with a low propionate level in the stool sample before Chlorella consumption actually shows the increase of the level of the propionate. And some people also increase the butyrate, and which means that Chlorella extract would have potential to increase the short-chain fatty acid in the gut. So I think I explained about this scheme, but it may be possible, not all the people, but in some people, that the immune modulation will also to induce beneficial bacteria could be achieved by the Chlorella extract. So this is a summary. The Chlorella heteropolysaccharide could be an active component in immune modulation or immune stimulation. So we need to think about how to use beneficially in the future, and also now perhaps. And Chlorella heteropolysaccharide used as prebiotics to increase the beneficial microbiome. And the function of that Chlorella and also each Chlorella species need to be investigated, because now we know that the different species could have different layer and different carbohydrate profile. Thank you very much.
Okay.
And oh, I forgot, this is acknowledgement. And Jishima's analysis and also lactobacillus acidus was done by Professor Oki Higuchi at Tohoku University. Thank you.
Okay.